Welcome to Nikolai's genetics lessons. In my 5 previous videos, I explained how to understand, read, and interpret genome-wide association studies results, the tables here on the left and on the right. And sometimes you also can see pictures like this below these graphs. So today we are going to talk about linkage disequilibrium blocks or triangles. They are also called LD maps, LD blocks, or LD triangles. Let me start with a theory. Imagine that this is chromosome number 1, or it can be chromosome number 2, whatever. Let's say this is chromosome number 2. And both these two chromosomes are actually homologous chromosomes. One you got from the mother side, another from the father side. So this is almost identical chromosomes, and the only difference between them would be some point mutations. Sometimes it can be deletions, insertions, and so on. Also, let's say somewhere in the middle you have centromere. Let's say somewhere here you have gene A, and on the homologous chromosome we have also gene A, but it would be slightly different version of that gene, so we call this allele. And let's say this is going to be small a. And also on the other tip we have gene B, and on the homologous chromosome also slight different version of this gene, which we will call B small. During meiosis, these two homologous chromosomes may have crossing over anywhere along the length of the chromosomes. So result would be following combinations. For example, it can be, I just going to show you smaller version. So it can be, for example, one variant, it would be capital A and capital B, small a and small b. Just this chromosome and this chromosome were inherited by the gamete intact. But also crossing over may happen. So other variants can be, other two variants would be recombinant. And it can be, for example, capital A and small b, or it can be small a and capital B. Result of crossing over: capital A and small b, and small a and capital B. If gene A and B are far away from each other, then we expect that crossing over would usually happen between them. If it's going to be single crossing over, we are going to have hybrids: capital A and small b, small a and capital B. If two crossing over events would happen, we are going to have small a and again small b. So only odd number of crossing over would give us hybrids, and even number of crossing over would give us the same variant: small a, small b, and capital A and capital B. So in gametes we expect that 25% would be this intact chromosomes if crossing over happened even number or didn't happen at all. And we also expect 25% of these hybrids and 25% of these hybrids. For us, it's going to look like independent assortment, as if gene A and B would be not on the same chromosome, but on different chromosomes. Now imagine different variant. For example, gene B is going to be much closer to the gene A. So capital B and small b here. As you see, when two genes are close to each other, probability of crossing over is going to be greatly reduced. Now we're probably going to have 40% of parental variant, which is not result of crossing over, another 40% is going to be a parental variant small a, small b, and here we are going to have 10% and 10% here. So together they are going to make the same 100%, just like in this case. But now we call that these two genes, A and B, are linked, meaning on the same chromosome and somewhere close to each other. And this is what we call linkage disequilibrium, when two genes would be linked and not going to show numbers like this, but analyzing these numbers, we can say that these two genes are close to each other, that they are linked and in linkage disequilibrium. Now we are ready to talk and analyze these blocks or triangles. Let's first concentrate on the upper part. The blue line here represents a fragment of the chromosome, and white bars on this blue fragment of the chromosome represent single nucleotide polymorphisms, or SNPs. These SNPs have been identified and sequenced, and this is positions where me and you can be different. 99.9% of my genome and your genome is identical, but in these places it can be different. This is what makes us unique, and this also true for any animal, plant, or any living organism, with exception of identical twins and those organisms which can be propagated by cloning. So as you see, each such position or locus, multiple we call loci, has its own unique name. These names come from database. As you see, all of them start with RS, plus also unique number which identify each such position. But here in this line, as you see, each of them would be represented by number 1, 2, 3, and so on. This is purely done for convenience. Now let me open the whole picture. What you can see here is block number 1, and the length of this block is 49 kilobases, 49,000 nucleotides. Here's a block number 2, which length is 23,000 bases, or 23 kilobases. Block number 3 and block number 4. And you also can see the length of each block. Now let's talk about the pattern. You see that some squares here have solid color and no numbers, some have numbers, and they also are different in the coloration. Now let's take, for example, we want to compare locus 7 and locus 11. Here we see number 87. What does it mean? In a probability theory, we usually work not between 0 and 100, but we work between 0 and 1. All our numbers are between 0 and 1. And for example, 87 actually means 0.87, but for our convenience, times 100. So all these numbers are a result of multiplying by 100. This is made for purely for convenience, because, for example, for us it's much easier to compare, let's say, some example, 87 apples and 79 apples instead of 0.87 and 0.79. So when we got 1 in our table, that means 100. So 100% that two loci would be inherited together. For example, locus 8 and 10. We do not put a number here. We just leave this square solid, but it's actually means 1 or 100. And this is highest number, and that means that these two loci in linkage disequilibrium and 100 also means that they would be inherited together, and usually no crossing over between locus 8 and locus 10 happen. Now some of you may think how we can get so low numbers. For example, 87 or 100 can be, as you see, explained that between two loci is a linkage. But how we can get numbers which is less than 25%, as in my previous example in the previous slide, which I show you, where you can get 25% in independent assortment, where two genes completely unrelated and on the different chromosomes. So how we can get less than 25%? The difference that these numbers we got as not result of single meiotic event or single generation, but across multiple generations, maybe thousands of generations, thousands of years. Specialized software programs already have this frequency, so you don't have to worry about it. And actually, such white blocks means that here, in these positions, between, say, block 1 and block 2, these white squares specify between locus 1, 2, and 5 and 6, we have crossing over hotspot. So here's one more conceptual idea for you. Crossing over happens not evenly across the whole length of the chromosomes. In some places it can happen much more frequent than in other places. For example, crossing over happens much more frequently in intergenic space between genes than within genes. But if it happens within genes, even within genes, as you know, there are spaces which we call introns and exons. So it happens more frequently in introns, non-coding sequence, between coding sequences. So you understand that if crossing over is going to happen within a coding sequence, there are more chances that some kind of disruption may happen, mutation within a gene, which would affect the health of the organism if it is important protein, if this gene code for important protein. So blocks are haplotypes, specify haplotypes, and within blocks we can find different single nucleotide polymorphisms. So if we know that these two single nucleotide polymorphisms are usually inherited together, that means that the whole sequence between them is also would be intact when would be inherited by progeny. I don't like to make videos longer than 15 minutes, so you would be able to digest information, and I probably would make another video on this topic. But before I will finish, these white squares represent 0 or hot spots for crossing over, and solid red colored squares represent complete linkage, or 1 or 100%. That SNPs would be inherited together. And this is all for today. Subscribe and see you in the next video. Goodbye.